So, good evening, Dr. Blackburn, dear Elizabeth, dear colleagues, dear guests. I guess some of you are here tonight because you would like to learn what you can do against the feeling of being stressed. Well, the answer is in fact magic, because it lies in the word itself. Just read it backwards. Unfortunately, it's not that easy. That's why we have founded Stressnetwork.ch and invited Dr. Blackburn for tonight's lecture. For those who are not familiar with the network, a few words about it. So it's a newly founded network of scientists from Swiss universities working in different areas of stress, such as psychology, psychiatry, economy, neuroscience, internal medicine, or genetics, and we have now 40 labs that are members of the network. You see actually here on the right side there is nothing, so it's basically throughout Switzerland, but only half of Switzerland, because on that side we have our beautiful mountains. And because the mountains are so beautiful, people are not stressed and are not part of the network. So why the stress network? Studies have shown that more than 25% of the Swiss population suffers from stress, from too much stress actually. And stress can lead to severe mental and physical health problems, and stress causes an economic burden of several billion Swiss francs per year. So we believe that a better interdisciplinary understanding of the complex phenomenon of stress is a prerequisite to prevent and treat stress-related health problems. So what do? We foster scientific interaction between different areas of stress research. So the whole day today we have had a scientific discussion with all different kind of researchers from different fields. And we would like to raise public and political awareness for the importance of stress research. So these are actually happening today. Now I'm very happy to introduce our keynote speaker, Dr. Elizabeth Blackburn. Dr. Blackburn was born on the beautiful island of Tasmania, which belongs to Australia. Do you remember this area there? Absolutely. Then Dr. Blackburn studied biochemistry in Melbourne, received her PhD from Cambridge University, worked at Yale University, UC Berkeley, Salk Institute, and now at the University of California, San Francisco. Here is a photo from her early career stage. It was taken on the first school day together with her older sister. I would spend now at least an hour by mentioning all achievements and honors of Dr. Blackburn, so I just highlight three. In 2009, Dr. Blackburn was awarded with the Nobel Prize in Physiology or Medicine for the discovery of how chromosomes are protected by telomeres and the enzyme telomerase. We will hear more about this discovery in her lecture. In 2006 and 2007, Dr. Blackburn was named one of Time magazine's 100 most influential people. And in 2001, Dr. Blackburn was appointed as member of the US President's Council on Bioethics. But the real achievement here is actually not the appointment, but the reason for her dismissal. Dr. Blackburn said, "I felt a strong base of scientific fact and evidence would be particularly important. I publicly made clear my views on some of the Council's recommendations, views that did not generally accord with those of the White House or those of the Council's chair." After 2 years, Dr. Blackburn was informed by the personnel office of the George W. Bush White House that she would no longer be on the Council. So we have learned an important lesson from Dr. Blackburn. Whatever it costs, always stick to scientific facts, never to alternative facts. Now, dear Elizabeth, We are very much looking forward to your talk.
Thank you so much, Dominic, for this invitation, and I'm just delighted to be here to talk about the relationships between stress and the subject which has been my research subject for more decades than I think probably about 4 decades. But particularly I want to kind of open out into the implications that this has for human health. And it's certainly been very eye-opening for me to take this journey. So what I'm going to do is talk about the science of telomeres, and don't worry, it'll be pretty brief, but there's a lot that we know very, very solidly. And then how I and then many others have got to link that kind of biology going on in us humans to the subject of Dominic's fantastic symposium today, which is all about the kind of stress which is not good. The good kind is the kind that we have standing up here on stage and a few minutes of, challenge. That's fine. But I'm talking about very severe stress, which can have toxic effects. And then it got me really thinking about broader implications. So, stress. The idea is not new that stress would be related to something which my research had taken me into over the decades, and that was the idea of aging. And so you may not know him, but he was a military general about 2,500 years ago, and he was crossing from one state of China to another, and he knew the border crossing was going to be really tight. They would try and capture him, and so the story goes that the general was incredibly worried about this, and with the worry, his hair turned white overnight, so you can see his whitened hair in the statue, and his features age, and he was able to cross the border because they didn't recognize him as a result of these physiological changes. So the idea is that stress is not, and the connection that you age when you stress, when you're very severely stressed for long times. In this case, it was relatively acute, according to the story. But the idea of connecting something that we always think about much more as chronological, aging, and something that we think about in a different way, stress, that hasn't exactly been a new idea. So it's important to us because, of course, we are, and Switzerland is certainly no exception, worldwide an aging demographic. And we know that there's a tremendous combination of factors of which non-genetic and genetic ones interact, and the non-genetic kinds of effects certainly can be put under various kinds of headings such as life events and behavior and environmental, which cover a lot of different areas. But they're not simply genetic, but clearly there's opportunities for interaction. And what's really interesting is that we all know that many of the common diseases, many of which cause morbidities which lead to then mortality, many of those common kinds of conditions listed here are often going hand in hand. So is there some commonality between things that on the face of it looked very different? Cardiovascular disease was put in a different bucket for a long time from diabetes, from things like mental or cognitive disorders. They were all kind of put in very separate clinical ways of thinking, but what's fascinating is that they often go hand in hand, meaning that one is often a risk factor for the other. So was there anything behind it? So now enter the research that I did for many years and got very interested in the ends of chromosomes, which are, at every end there's about 10,000 DNA building blocks made up of, when you were born, of this very simple repeated DNA sequences, and they form a kind of a scaffold upon which a protective sheath of proteins, very well characterized, sits and protects the ends of the chromosomes from all sorts of things that could really mess up our genetic material if they happened unchecked, like degradation from the ends or the ends sticking to each other, preventing chromosomes from separating properly at division, and so forth. So this is an important part of protecting our genetic information. But the key point is that there's an age-related phenomenon in humans which is observable, and that is that on average, I emphasize that, as we get older, those tracts of telomeric DNA on average are liable to get shorter and shorter. It's for a variety of reasons. And it's not as if it's unchecked at all the time, because there is an enzyme that we discovered in the 80s, which is called telomerase, and it has the capacity to elongate telomeres, adds DNA back so the sheath can be built back up bigger. And yet, despite that, existence of such an enzyme, in us humans, it's often very limiting. Normally, in us in the room here, we as a group will be showing average telomere shortening over the decades as we age. And so even though many of our cells have telomerase, it's highly regulated down in almost all cell types. Luckily not in our germ cell lineages, luckily, and sadly, it's not downregulated in cancer cells, but it is downregulated very much in a lot of cells, not to zero, but to downregulated. And so it's a complex set of regulations that go on. But the interesting thing is that, as I said, shortening happens, and I'll just list a few ways that the shortening is known to happen. DNA can't be replicated completely all the way to the ends by the cellular DNA replication machinery. So every time the DNA replicates, it actually leaves some of the chromosomal end unreplicated. Telomerase can make up and replenish that difference. But it's also realized recently that even cells that don't divide much at all can have telomere shortening events, which are nothing to do with this replication. Often it's chemical oxidative damage. Replication moving to the telomere end can get stalled off and the ends can break off. So telomeres can abruptly shorten even in cell compartments that are not highly proliferative. This is relatively recent knowledge, and I think it broadens, our understanding of what might be going on with us, because, as I said, there's this general observation of telomeres shortening. By the way, you don't have to shorten them down to nothing, about a little bit under 4,000 base pairs. That's getting to a point where the telomeres just can't protect, and now they're unprotected if they just fall a little bit below 4,000 base pairs. And that's just not enough to keep a stable end. And now that DNA looks like a broken chromosome. Cells don't like that. They have a very clear set of responses, and they persistently signal from these broken, now seemingly broken ends. The cell reads that as I've got a broken DNA, and it starts signaling alarms, but the telomere doesn't have a normal way of fixing that. A broken end inside a chromosome gets sealed back again by molecular mechanisms. So this is the key of the problem for cells if telomeres fall below too much of, fall below this critical limit. And a list of things happens, and I'm going to allude to these, but it's well characterized that in cells, the cells cease dividing, they become what's called senescent. A whole program gets reset up whereby they start sending out alarm signals, including pro-inflammatory, pro-tumorigenic factors. So the cells have stopped multiplying at that point, they've arrested, so cell renewal capacity has gone down. Other functions, such as the mitochondria, the energy powerhouses of the cells, as a part of this DNA signaling, this broken DNA signaling, mitochondria are getting signals that prevent them from functioning properly. So this is what happens in cells. And as if that wasn't bad enough, there's also, because of what happens, a sort of a vicious cycle can get it set up, both affecting the same cell where the telomere is shortening and other cells. So let me just walk you through this. So here's a telomere, and the idea is that it's going through shortening. Let's say that cell's been dividing, or the cell has suffered oxidative damage of some kind, and now that becomes an uncapped telomere, and it starts signaling. It reprograms the genetic material in the cell, the transcriptome as it's called, and one of the effects is it signals to the mitochondria, the energy powerhouses of the cells. They're not properly regulated. They produce reactive oxygen species. This, as I mentioned, telomeres can be damaged by oxidative damage. They're actually very susceptible to it. You also get secretion. So now the factors can start affecting other cells. So this reactive oxygen species, in turn, now can further shorten the telomeres. And now, this process gets emphasized, and now a pro-inflammatory environment physiologically now also increases oxidative stress. So you now got another source of oxidative stress. So you've got a vicious cycle set up. So the short telomeres can set up a set of situations in the cell and outside the cell, which then provokes even more shortening. So this has all been quite well studied at the cellular and to some degree organismal level in model organisms. And of course, the process of aging has been studied in a great many model organisms and of course is being studied in humans. And I want to relate this to a lot of very, very good science that's gone on with aging. Now, aging in all sorts of organisms, including those that are quite short-lived and for whom telomere shortening, unlike in our decades of life, is not a feature of their life cycles normally, but aging has a lot of parameters that have been seen at the cell level. Stem cells, which replenish tissues, get exhausted, right? So they fail to replenish tissues. Cells, you can find cells that have those senescent phenotypes. One of the reasons is telomere shortening, but it's by no means the only reason cells can become senescent. If you look in organisms, you find other things. Mitochondria, metabolic dysfunction. These things happen with aging of many short-lived organisms too. Inflammatory states get worse. One finds long-lived proteins suffer damage over the lifespan of organisms. So you have all these things that go on, absolutely seen very clearly in organisms in which telomere shortening is not apparently very relevant. But now in humans, you have this overaging, you have this attrition. And what's particularly fascinating is that this has the potential to drive or promote all of those features that I just listed, which have been seen in aging models over and over again. And it hinges on this persistent DNA damage response. And that then sets up a series of things which have been individually quite well characterized. And so I'll just list them all. The cell senescence, you get cell cycle arrest. Okay, if cells are not replicating, if you're a stem cell, that means the stem cells are no longer forming new tissues easily or effectively. The response, so you get this observation. So this can lie downstream of that telomere attrition leading to the persistent DNA damage response. The cell senescence pathway, as I mentioned, can certainly lead to cells not replicating anymore. And as I mentioned, mitochondrial and metabolic dysfunction, and there's a particular pathway of transcriptional control of this that has been well characterized. As I said, pro-inflammatory secreted factors. And then those two together, you're increasing the reactive oxygen species levels within cells and also organismically, and that certainly is one way you can damage long-lived proteins. So now you've got a sort of hierarchy where telomere shortening can be at the top. Each of those brown arrows has been individually looked at, but what we don't know is, if we look in humans, how important are the relative sizes, if you will, of all of these arrows. Individually, they all can lie downstream of telomere attrition. But the only way to find out in humans, what's going on with telomeres is to do measures in humans. So you need to measure telomere lengths. And we and others have done this very extensively. And for many of the studies, we've used a really well-characterized, carefully done method where we just measure average telomere length in cell populations. And that's really because it's most practical in human studies, especially as I'll show you later when we have 100,000 people we look at. It's not easy to be analyzing telomeres under the microscope, every single telomere in the cells, although we do that in the lab and many others do that for research. But we measure average telomere length. So it's heterogeneous, but as telomeres' average length shortens, the likelihood goes up that they will fall below that critical level. So we measure it in actual humans. Now, this is really for the cognoscenti. For those of you who are a little sleepy can drop off to sleep a little. I just want to show you something, this is getting down and dirty, but people always say, well, do telomeres shorten at similar rates in different cell types in the body? So the answer is no. But what is very interesting is that you can see two kinds of things going on. And one is that if you look at telomeres, say, in sorted immune cells in your peripheral system, white blood cells you get from a standard blood draw from healthy individuals will give you this as well, then you can look at the different kinds of cells and you can analyze telomeres and you can analyze them over time as well. And what we find is that the different subtypes of cells, they have distinctive rates of shortening. They have distinctive rates, average telomere lengths, but they do correlate within an individual. So if all of your telomeres in your CD4 cells are long, then they're going to be long in your PBMCs, the peripheral mononuclear cells, also your leukocytes, even your salivary cells. Chances are high. But there are cell type specific differences. And you can even just take the whole mixture of simple prep of peripheral blood mononuclear cells and just analyze that, because that's practical. Costs and getting samples can be limiting, so we are looking at mixtures of cells. Now, the molecular biology, nerd in me just hates that, but in fact, it turns out to be quite informative to be looking at just these mixtures of cells because they're all responding in systemic ways as well as cell type specific ways. They all correlate quite well. So that means we can actually look, even though different immune cell types do have different average telomere lengths, they do have different average rates of telomere change, but they go together in a correlated way in one person. So if one cell type is changing in you, it's changing in another cell type will be changing in you at a certain correlated rate. In another person, that will be a different correlated rate. And so that means that not only is there differences, and so that's fascinating biology behind that too, but they're correlated. And so I'm emphasizing this because a lot of the studies we do in humans can, just for practical reasons, be best done with things like peripheral blood mononuclear cells, sometimes just leukocytes, white blood cells, and even saliva, even more of a mixture. But we clearly see correlations within a person. So you get a rough measure, a rough window into a person by using samples, even though they're mixtures of cell types. So I've taken a bit of a long time to emphasize this, but just to tell you, we have looked at it carefully, but the bottom line is you can extract information when you look at cohorts or populations of people, even using mixtures of cell types. And so this is what you see. As I said, telomeres roughly shorten with age. And so there'll be two things that strike you. This is just a typical study that was done with a few hundred people, years ago. And basically the x-axis is increasing age. And you can see there's a general trend downwards. But it won't escape your notice how extraordinarily variable telomere length is at any one age. I mean, if you wanted to find out your age, this is certainly not the way to find it out, right? But the question is there information in there? That's the interesting question, and that's where the research got interesting in humans. Now, first of all, you want to say, well, if this is just an observation that telomeres get shorter, really, does it matter if telomeres are shorter or not? And what was very helpful, starting in about 2001, where Valiami and Dokal et al. In London and various other clinical genetic centers found that there are mutations in the gene that encodes part of telomerase, very clear function, and those mutations cause phenotypes, even when you've only got one mutant copy out of the two copies of that gene, one from your mother, one from your father, and you only have to have one bad, still got one good, and the phenotypes are very dramatic. And this is summarized in this rather busy slide, but you're showing a lot of things here. So a child comes in, and they have a set of phenotypes, which has a strange name, dyskeratosis congenita, but it's a lot of phenotypes, including aplastic anemia, gastrointestinal issues. A lot of different kinds of rather bad phenotypes are going on. I'll list them in a moment. And then they look at the family history and they find that the parent has also had an interesting history. They've had often aplastic anemia as a prominent example. And then you go to the grandparent and you find the grandparent also had an interesting set of things, often manifested, for example, as pulmonary fibrosis. These are very different looking diseases, but it's one mutation being passed down. Now, the curved lines are just the percentiles for the telomere length, just like that scatter plot I showed you, just drawn for populations, normal populations. So you see that the telomere lengths are falling lower for this set of, this is roughly what you would see, where the telomeres get shorter and shorter as you go down the generations of the people carrying the mutant in this families. And there's literally hundreds of such children now where these have been found and put in family categories and pedigrees and so forth. So basically, when you have these really short telomeres, there's a very severe phenotype, but you've only lost half the gene dosage of the relevant genes. There's about a dozen such genes. And interestingly, almost all of them are very well defined as to what they do, and many of them are involved directly in telomerase action. And so they're very well dedicated genes. So this looks very causal, right? So and what sorts of phenotypes have been looked at? The penetrance varies, which is fascinating in these families, but this whole list of things has been seen en masse, as you look at all of these patients, different ages, and what you'll recognize is that there's a tremendous overlap with the normal phenotypes of normal population aging. The difference here is that they're faster, and their onsets are faster, and they're often lethal. So by the time the children have inherited for a couple of generations, they often don't make it into much beyond, adulthood or middle age. So this is serious sets of things, but the nature of them all heavily overlaps with typical sorts of things that you see in variable ways all over humans as they age normally. So you can say, okay, well, that's an extreme case. The mutations are extreme. What about in the population at large? Now, two kinds of things, which I'm going to summarize, a lot of different cohorts and a lot of people's work, but essentially you can measure telomere shortness and you can correlate it with not having the disease, although you can do that case control, but will somebody get the disease in the future? And the shortness increasingly predicts the likelihood, the frequency with which in cohorts you will see a whole series of diseases which overlap with those of the extreme ones. And many of them actually account for mortality as well. I'm going to talk more about mortality. But so that's a correlation. Doesn't say which way round it was, although it's interesting, the shorter telomeres did precede the onset of the diseases, but of course a lot of these disease processes are unfolding very slowly subclinically for a while anyway. So this says nothing about directionality. But genetics does. And so in recent years, big GWAS studies have been done now in normal populations, not populations with frank diseases, although you can see correlations, I'll show you. And two very striking ones I'll just highlight were when you look at the common variants of genes that we know are dedicated to telomere maintenance, because we know their molecular properties and their functions very well. Two actually come out very clearly. There may be more, but I'm just highlighting two, where the data is really nice that if you just have a handful of common variants that go along with slightly shorter telomeres versus slightly longer telomeres, treating telomeres as a quantitative trait, as so many continuous sorts of complex traits are in humans, there's really only a handful of genes that have effects you can discern in GWAS studies. Gratifyingly, they are telomere maintenance genes, telomerase components, or those protective proteins on the telomeres. And basically, it's common variants of known telomere maintenance genes that cause you to have slightly shorter telomeres. Now, they go along with higher risks, lifetime risks, of cardiovascular disease of various manifestations in an allele additive way. So it's a really nice quantitative relationship that you can discern. Cod et al. Did these studies with tens of thousands of people. Others have done it. And also, interestingly, dementia's Alzheimer's was the one that showed up. Quite decent sort of effect sizes. So now, I think most rational people really think if you have good Mendelian randomization, you can really attribute functions to genes, then there's causality there. Okay, so there are effects of genes. They're not that huge. I mean, for the top 6 or 7 genes that are the ones associated with shorter telomeres in an additive way and increase the risks of cardiovascular disease, if you have the short alleles for all of those, then your risk goes up about 20%. So, but still, that's not nothing. But of course, most people don't have all of the short alleles of all of these genes. Remember, these are normal variations here. These are not pathological variations in the sense of the single gene diseases I talked about. And mortality also is anticipated. I'm going to talk you through a bit of that data soon. But now we want to get to things that are not genetic, because many of those kinds of diseases that I listed, immediately you're looking at them and seeing cardiovascular, diabetes, and so forth. You know that many of those are not simply genetically determined. They are clearly influenced by life-related aspects. And an interesting one is chronic psychological stress. A lot of good epidemiological data link chronic, often severe psychological stress of one kind or another, and we'll get into the kinds, with increased risk of, for example, heart disease has been particularly very good large studies, at least at the time when we started these studies, which I started with a colleague who's a psychology researcher at UCSF. Her name's Alyssa Epple. She's a professor in the Department of Psychiatry at University of California, San Francisco. And in my lab group, I want to highlight Jolyn, who really has led for the last almost 20 years these human-related studies in my lab, where we do telomere measures in all sorts of interesting studies in humans. And Alyssa came by, it was probably 15 years ago, and said, people who are stressed out, they clearly have these sort of phenotypes, they look older and so on. Does anybody know what's happened to their telomeres? I think she thought, well, of course, everybody must have looked at this. I said, no, it's a really interesting question. And so that started a whole series of first collaborations, and then many other groups have done many more studies. And I'd like to just highlight a few. So our first study, so she said, was telomere maintenance affected by severe chronic stress? And of course, the implicit question behind that would be then, might that be something that is related to the risks of these very common diseases that are such common causes of morbidity, often comorbidity in humans as we age? So the first thing was to ask that, and so she had a very well-studied cohort of highly stressed women, premenopausal. They're the mother of a child, the biological child, who has a chronic condition of some kind or another, just a lot of different kinds of possible conditions, and they were the primary caregiver. And I point out in the US, very often that means you don't get much social support, from any kind of organization or state, or the health. You're not well supported commonly in the US. So this mother bears a burden. So this has been often used as a good sort of studying naturalistic experiment situation where you can look at somebody who's under prolonged and severe psychological stress. And so what was interesting was that even after we corrected for the various sorts of things like age and so forth, as you should, there was this relationship that just popped out between the years she had been in that situation, the shorter were her telomeres. So that was the first thing that popped out, and wow, it said there's something, either it's never going to reproduce again, or there's something there. So, and some other measures of, perceived stress and so on correlated with telomere shortness as well. But this one suggested that the stress was causing it, because it was harder to imagine that the shorter her telomeres were, the more likely she would be having an increasing number of years of exposure to this, right? So you have to, think about models, could be possible. So then another cohort was another kind of caregiver, again, very long-term stressed, where now these are postmenopausal women, and they had been the primary, again, caregiver for their spouse, usually their husband or a family member with dementia. So again, a known long-term stressful situation. And so all sorts of interesting measures of stress that the psychologists use, like perceived stress level, pessimism, interestingly more a trait-like long-term characteristic, eating disorders, distressed eating disorder behaviors, anticipatory threat appraisals, setting up in one of a known stressful lab situations. And again, you just kept seeing this same kind of relationship. So, you know, these are small studies, but well-controlled and well-done studies. So there's something was there, right? And so that then really sort of went into a lot of different directions with different groups, and now you can look at people in different life situations. This is one that maybe especially relevant for Brexit. I hadn't thought about this, but this was the Whitehall study of civil servants, and on measures of cynical hostility, I kid you not, and carefully controlled for other variables, there was the relationship with high cynical hostility and shorter telomeres. And again, these were carefully done studies where, you know, variables were carefully controlled. On the happier side, in studies we looked at healthy women, middle-aged women, and found that there was a nice relationship between the better sleeping habits they had. I think this was a 3-month self-report, the longer were their telomeres. And even in studies where you can start trying to kind of affect your sort of attentional state, for example, measured by mind-wandering tendency, the better your attentional state could be, you know, with decent number, it was well-done studies, you saw this sort of graded response, longer telomeres. So I could go on, but I just want to highlight something that will take me into more thinking about, you know, what are some of these broader implications of what we're finding. And that is a different kind of stress, and this is a stress where now the person really has had very little control over it, and this is childhood trauma exposure. And if you simply look at the number of different categories of childhood trauma, this is self-report, and these are young to middle-aged adults now, so this is years behind them often. But if they're exposed to not 1, not 2, but 3, but 4, 5 different categories, there was this sort of graded response in the sense of the telomeres were roughly dose-dependent, shorter and shorter. Interestingly, this group here also had post-traumatic stress disorder, but that turned out not to be the primary determinant. It was the primary determinant was childhood trauma exposure. So now we're looking at something where, you know, there's much, you know, there's societal issues here at play here. And grimly, this kind of thing has been reproduced looking at childhood adversity in different ways over and over, and cumulative exposure to traumatic events. It's really associated with short telomere length in when you do age and other adjusted telomere length. And this was a meta-analysis. I'll just show you, if you can read you know, it's just a grim set of things that these children were exposed to. But the average age in which telomeres were measured, and the likelihood you'd have shorter telomeres is off on this side here, was about 42 in these meta-analysis. So these are people well into adulthood. And yet their telomeres, if you look at these studies overall, you conclude from this meta-analysis that things happening, severe kinds of exposures to children, had an effect that you could quantitatively read out as telomere shortness. And why does this matter? Well, remember that there are these clear studies where having shorter telomeres quantitatively makes you more likely to have one of the major diseases of aging, you know, such as cardiovascular diabetes, certain cancers, immune dysfunction, lung dysfunction, a whole lot of things that are age-related and quite, you know, severe morbidities. So you might say, well, what's stress got to do with telomeres? And my collaborator David Spiegel at Stanford, who's a psychiatrist and looks at stress often in patients who've had cancers, but this is just general, put this nice schematic together. And again, each of these arrows connecting the brain, which is clearly where the stress signaling will be both inputted and then will be then played out physiologically. And each of these arrows, has been well documented. Yes, when you have prolonged chronic stress, you will have dysregulation of cortisol, diurnal rhythms and levels overall. We know that very directly, we know it decreases telomerase, therefore telomere maintenance in normal immune cells. You also set up other glucocorticoid, among others, mediated suppression of certain DNA damage response genes, which in turn will all play into the likelihood of telomeres being shorter. When your immune system's telomeres get too short, one of the things that happens is that the cells can't respond and proliferate properly, so they go into immune senescence. And then, as I mentioned, you get this pro-inflammatory situation, and the sort of cycle can get set up. So it's not irrational or even that mysterious now that here's at least one pathway by which shorter telomeres certainly rationally can be seen to be a consequence of what goes on physiologically in humans under chronic stress. I don't want to say this is the only thing by any means. There are probably other ways that the telomeres are being impacted, and I certainly don't want to imply that this is the only thing that stress is doing, but it is one that, our lens has sort of been on this one because you can quantify it, and it is so well understood at the cell level, this telomere attrition and then loss of telomere proper function. Okay, so I said I would talk a bit about mortality, because you say, well, this is all fine, but what about death? That's a pretty clear phenotype, and that sort of matters, right? You could put up with your type 2 diabetes and so forth, but if it kills you, then serious, right? So a few years ago, we got some funding. About 8 years ago, we got some funding. We set up a big setup to automatically measure telomeres in 100,000 people, which came from a big cohort, which was like a big health system in Northern California. It's called the Kaiser system, and they got 100,000 people, more than that, to agree to have their genotypes and their telomeres and all sorts of survey and clinical records and so on included in big studies. It's all being deposited in the National Institutes of Health DB gap. And so a lot of this didn't end up getting published yet, but it's all being deposited there. So anyway, we set this big automated system up, and we said, what happens with age? And so this is the means. And so what's interesting is that you see the decline, but then at about 70-ish, which is the maximum rate of mortality age group in this population, you get the lowest means, and then it's more and more likely that if you lived longer and longer, your telomeres actually were longer and longer. Now, we don't think that telomeres went down and then went up. This is all cross-sectional, remember. And a very nice hint came from another study done in Costa Rica, where there's a peninsula called the Nicoya Peninsula. And in the Nicoya Peninsula, it's famous, it's called a blue zone, because people often live to 100 more often than in the population at large in Costa Rica. And so we, with David Rieckhoff and others, did analyses of telomere lengths. And so the lines and the shades show the means again. First of all, you can see the enormous variability with age, just as I showed you before. For any one age, each dot is an individual subject here. But right away, you can see from the lines that, from middle age onward, these people have had longer telomeres in the long-lived Nicoya population than their counterparts out in Costa Rica at large, controlled subject groups, and then they converge when people get to around 100. So I think what we think is happening is that in this 100,000 people we looked at, that the telomeres, through their life have probably on average been longer than their counterparts who are the more common situation in the population. As I said, maximum mortality is somewhere in the 70s in that population. Okay, so now with 100,000, we could play with all sorts of numbers and make sure that we were seeing things that others had seen in other studies. So dose response for cigarette smoking history, dose response for another very interesting parameter, which now I think strongly relates to stress again. And again, takes us into sort of broader areas, and that is what kind of neighborhood do people live in? So there's a wide range, as you know, in the United States, and this area covers a lot of urban areas as well as suburban areas. And so the Neighborhood Deprivation Index, which is about 8 nationally agreed on parameters for essentially neighborhood horribleness. And so you have an index, and so they were divided into quartiles of neighborhood horribleness. Right? I mean, it sounds trivial, but this is not light-hearted stuff. And so here you could see, first of all, just if you simply adjusted for age and sex, then because there are gender differences in telomere lengths at different ages, you could adjust for those, and you can see the red triangles, you know, compared with the best neighborhood, where it's set to 0, the deviation downwards from it, you know, follows somewhat a trend, right? And so then you can say, well, you know, but a lot of things are going on. So why don't we add, you know, all other things that we know, we see subtle changes, you know, race, smoking, obesity, education, I'll show you, income, you know, put all these in and barely, and then add in, well, you know, what about cardiovascular disease? What about diabetes? That barely shifts it. So in other words, this effect of being in your neighborhood seems pretty strongly not able to be just foisted off on, well, they just had more diabetes, or they just had less income, or they, you know, something like that. It looks rather like the neighborhood itself is having some, you know, relationship with telomere shortness. And other studies have certainly shown comparable things here, but this was done with a very, you know, with a large group of people. Another very long-lasting thing. So right away, we're now not in you know, the world of medicine or biological research. We're in the world of socio-economic issues. We're in policies. We're in much bigger territory now than just looking at an individual's you know, medical history or something like that, because we're looking at the world they're living in. And so I was really, you know, I think this is striking. Another striking thing came again first from the Whitehall study I referred to you, this long longitudinal study that Michael Marmot and others have been studying in London, University College London people. And they showed some years ago a nice relationship with telomere shortness and inability to have completed what in the US you would call high school, right, secondary education. And again, they said, well, of course, you know, and these were middle-aged to elderly adults. So this is way back, you know, when they were much younger, this, whether they did or didn't have increasing you know, ability to finish high school or then go on to university of some kind or another. And so they said, well, you know, of course, maybe this is accounted for by the fact that, you know, you have more education, you have better health things. When you've had more education, you have better income, you have better jobs. And so they corrected for as many of these things as they could. And as you can see, it barely shifts. It really doesn't seem to shift a whole lot. So here's middle-aged to elderly individuals where there's a sort of echoing of their educational, in this case more severely, the fact that they didn't finish secondary schooling, for which there can be many reasons. But again, we're coming to much bigger issues than simply, you know, somebody's simple brain power. I mean, most people are not failing to finish high school because they're stupid. There's things going on in their lives. And we echoed that with the 100,000 people too. People who didn't finish high school, same thing. They had lower telomeres, and it was a graded thing. So these are not huge effect sizes, but you can see them because the numbers are there. So Whitehall study and this Kaiser study, you know, population in the UK, population in Northern California, with quite a range of situations in life, you see the same kind of thing. So there's something going on. But as I said, okay, so now we know a lot of factors we can now, you know, correct for and ask questions about mortality. So this was the actual study in the 100,000 people. So we measured telomeres, and then the years went by, and this cohort, average age in the 60s when it started, but ranging from 20 to 95, you know, 3 years go by, and then you simply say who had died, and then you can say, well, how did that relate to, what was their telomere length like at the beginning? Did that have any bearing on who died within that 3-year space from any cause at all? And the answer was that there was a relationship, and the bottom quartile, which is set here, that's the shorter telomeres, you're about, 20% more likely to have died of any cause than the rest of the telomere length distribution people. And then you could say, well, that was because of all these other things. So we could correct for these effects that we could see of all these other factors. And you can see it barely shifted it. So it really looked like it's fairly, it's hanging in there as a pretty independent predictor. Very statistical. This is not something you would make a medical decision based on. But if you're making public health kinds of decisions, this is sort of the kind of information that I think it's very objective. So now you can say, well, it's just, crazy Californians, right? So now we'll go to the bastion of sanity, Scandinavia, right? And so now this is the Copenhagen study, and Rode et al. In 2015 did something which is very reminiscent. They have telomeres. They're now using leukocytes. We used saliva DNA, but as I say, they do correlate within individuals. So they looked at leukocytes, and they let about 7 years go by on average, and then they asked the same question: who had died? Nice big numbers, and so how did that relate to telomere shortness? And it was a really interesting thing. Now, they divided theirs into deciles of telomere length, and they get this beautifully graded response. So the top is just the raw number of deaths as a function of how short your telomeres were. And I've also plotted down here for deaths. So that was all cause, the red, but the blue is any kind of cancer. If you lump all the cancers together en masse, this was their finding. And so then you can look at other causes of death, and they have them all in good registries there. And so you could look and find death from cardiovascular disease, all sorts of other things. And you just kept seeing this sort of graded response. Now, again, this was raw data, so then they adjusted it. So if we look at overall deaths, you do see that there is some modification, but you still see this general trend. Okay, so this really is holding up that the shorter your telomeres, the more likely you are to die in some number of years. So this is looking real. Now there have been a lot more studies, and I just put all the words in here because essentially they're sort of converging on much the same thing. You're about 25% higher hazard of death comparing shortest to longest quartiles. We saw that. That roughly correlates with what the Copenhagen study saw. And interestingly, the effect is much less for people over 80. Remember, we find longer and longer telomeres seem to have been selected for people above the mid-70s. So you expect the relationship to go off the rails above that age. So this is also very reassuring. In other studies, we see this. So where are we getting at? The idea is we're just saying, look, telomeres are certainly a statistical predictor in a large cohort kind of way of mortality. It's not telling you much about you individually. Frankly, if you have your own telomeres measured, if you don't fall in that very, very low percentile, it's not going to tell you an awful lot. You have to be looking at cohorts here. But I'm going to tell you a little bit about how you can make that more precise in a moment. But one thing you can do is you can also look at rate of telomere shortening. So I've said, cross-sectionally you see this decline, but what if you look in a person? How fast do their telomeres decline? And this was very interesting in a cohort of people in San Francisco who had coronary heart disease stable, so-called. They were outpatients. And we looked at 5 years, and we said, from baseline to 5 years later, did your telomeres grow? Because they do grow. They can go up and down. Remember, there's telomerase in low amounts. You can get growth as well. Or did they shorten, or did they stay the same? And we just divided that into deciles. And then we said, okay, or our collaborators, Mary Hooley and colleagues at the Veterans Administration Hospital in San Francisco, did this. And these were not very healthy individuals, sort of in their 60s. And then they said, well, in the next 4 years, who died? Okay, as a function of how much their telomeres fell. And you got this graded relationship where you're at least twice as likely to die if your telomeres were shortening than if they were lengthening. And this is not trivial numbers here. Look at these percentages. It's going from about 20-some percent, less than 20% to over 40%. So this is not like just a few people dying. This is a lot of people who, in this pretty unhealthy cohort, are dying, and it's related to the rate of telomere shortening in these adults. And there's been discussion in the field of, well, how much do telomeres really shorten in adults? Well, in this sorts of situation, the shortening is quite predictive of mortality. In, well past, early adulthood, these are middle-aged to elderly people. Okay, now I just want to say a word about cancers, because I gave you an overall picture of cancer, but cancers I'm putting very advisedly in the plural. Some people count there might be 200 different kinds of cancers if you count all the different molecular pathways and organ sites and so forth. So we can't lump cancers all together. But in humans, they're unified by a very curious thing. As you go, from progression into cancers, telomerase becomes upregulated in something like 90% of human tumors, all different kinds. It's one of cancer's favorite things to do, is to upregulate telomerase in the fully malignant state. But what's really interesting is that if the system is a little bit primed by genes that make your telomerase just a bit active, that also increases your risks for certain cancers. So just to fill that out, what the situation is if you don't have enough telomerase, remember the rare genetic diseases I told you? They actually have an increased risk of this categories of cancers, hematological, squamous cell, gastrointestinal, often things that involve cells that are replicating fast. It might also be impairing immune surveillance, something not looked at yet. But the interesting thing was that human studies, and this, nobody asked for this, it emerged from analyzing genetics of cancers and genetics of people. Just a little bit more active telomerase, genetically determined, is enough to increase your risk for a whole other set of cancers, and they're prominently melanomas, brain, lung, certain liver cancers. Okay, so cancers are diverse with respect to this, so it's both ways. So essentially, what can we do, right? Right? There we are. That's the reality. But recall this, which is if you lump all cancers together, there is an overall likelihood of shorter telomeres being the ones that overall are the ones that are more likely to lead to death from the cause being any cancer. But that's very dependent on the cancers. And I just need to talk about that because this is nuanced in humans, as you can see with the cancers. It's pretty much one way for cardiovascular type 2 diabetes, for pulmonary fibrosis, and for various, certainly for cardiovascular disease in various manifestations. But the cancers are a bit more mixed, but overall there's that trend there. So now I want to just quickly go to, can this be any use to us aside from population studies? And a study out of the MD Anderson Cancer Center with about 440 bladder cancer patients really said that there's gold in this measure, which hasn't really been used to its maximum benefit because it hasn't been combined with other parameters. And a really interesting measure came out when they just looked at the time of diagnosis in bladder cancer patients. Now, and they combined two things that they had good reason to ask the question for. Telomere shortness, was that going to affect prognosis, later prognosis? And depression, and this was on a 32-point depression scale, just a simple median split for both depression scale and a median split for telomere length, long, short, in the white blood cells. Two very simple parameters taken at diagnosis. And now either one wasn't very predictive alone, but things don't happen in isolation. And in fact, both together were very predictive. Each one was pretty weak, but if you combined having shorter telomeres and worse on the scale of depression at diagnosis, that greatly increased the mortality. So that's the schematic. And I just want to show you, this is their data, their Kaplan-Meier survival curves. And this is just to show you that they really did very good. They controlled for all sorts of variables that you might expect would affect mortality, and when they controlled for it, they got very different survival curves. This is their data, but I want to show it to you more graphically because 441 people come into the clinic, they've got bladder cancer, they look at them on the depression scale, they look at them on telomere length, and then they wait. And of course, they're treating them and so forth. So here they are, 2.5 years later. If you just had short telomeres or just were depressed but had long telomeres, or neither. The black circles are those who've died in 2.5 years. And if you had both short telomeres and were depressed, this is the fraction at 2.5 years. So, it's already quite bad. So a lot of cancer, you wait until 5 years, because that's sort of the telling time. So now, if you had neither or just one of the other, of course, more people have died by 5 years. But if you had both, everybody had died. So this is really quite a striking result, because this now starts to get into territory where it starts to be clinically, I think, relevant. So just to make the point again, that this is an interaction here. And this is where I think there's such opportunity, because people really haven't looked at combining telomere measures with other things. Most of the studies have sorted it out and then looked at it by itself. But I think it interacts, and what's interesting is it's interacting with the whole system. This is the person. This is not the cancer's telomeres. This is white blood cell telomeres, and this is, a meta phenotype, if you will, depression. So together we can say it looks as if there really is a case to be made that properly regulated chromosome N maintenance does help protect. Conversely, if there's impacts on it, then those impacts can negatively affect the likelihood of getting all these comorbid diseases, often comorbid diseases of aging, and there's a lot of inputs into it, but of which ones that we have a lot of controls can matter. So all these things that are not genetic, there's a whole compendium of them now, and I've just listed them all here. So there's the bad ones, and then there's the good ones. Almost all of these to date have been observational studies, but some have now started to be much more, randomized control, prospective, proper studies. They're small, but they're still showing these same sorts of things in terms of improving telomere length. But what I want to highlight is the really quite striking lot of different things that are affecting telomere length negatively, of which I've listed a whole lot, and this is multiple studies again, and I've shown you a few examples of them. And I just want to end up talking about one of them, which is a very interesting question, because it has to do with our legacy to our next generations. So prenatal stress, it turns out, this is just one study that we did with Sonia Entriger and Pitik Wadwa at University of California, Irvine, and this is stress measured in the mother during her pregnancy, and then the telomere length in her newborn. And you can see that there's a relationship there. And in another study they did, where we looked at telomeres, this was in the 20-some-year-old daughters of a mother, and if she'd had a very severe stress during her pregnancy, those daughters had shorter telomeres, even 20 years later. So this is saying things that we knew already, that pregnancy conditions are very important, but now we can see it in this quantitative way, and it looks as if it's lasting. Now, when you think about transgenerational effects, what do you think about? You think about genes, right? You hand on genes and culture and so on. You also hand on the ends of your chromosomes directly through the sperm and the egg. They come through into the newborn, the conceptus, which then grows into a baby that's in childhood and so on. So the telomere length that they start off with turns out to be fairly influential in terms of predicting further telomere length changes, and also, as more and more data suggest, we know that things that happen prenatally are important. So one of the things that's, I think, quite unanswered is, in the population at large, do these other reasons that I've shown you are so many that can make telomeres shorter in a person, do they have enough size to, effect size to get passed on to the next generation? The genetic diseases, the haploinsufficiencies, very short telomeres for sure get passed on, and things get worse and worse with increasing generations. But that's more extreme. I think the really interesting question we have to ask is, are these non-genetically caused telomeres shortness in parents, are they handed on? Because we know they're handed on directly, but do they get adjusted for in the children or not? And we simply don't know the answer to that question. But to me, that's raising really big questions about how stress is influenced by all sorts of things, which is really impacting so many different aspects of our life as societies and as individuals, as, communities and so forth. There's all these sorts of things that clearly we, I think, have to start paying attention to. So it's really kind of opened my eyes. I used to look down microscopes. Now I look, I try and look out the window because there's so much going on now in terms of the things that impact telomeres. And that, in turn, we have very good reason to think that a population is impacting on human health and the converse risk diseases. Okay, so I've summarized here all of these things, the sort of sweep of what we have learned from this very basic science to collaborating and from many studies from many groups around the world, which is that yes, of course we do know lifestyle, mental health, environment. Of course, there's lots of good data out there, nothing to do with telomeres, that they're significant contributors to human health. But these kinds of things and severe chronic stress is certainly one measurable part of that in many, many ways. These impact telomere maintenance, and that in turn can be a way of quantifying how much they're contributing to these health outputs. And furthermore, we know that from genetics, we know that shortening of the telomere is not just a sort of bystander thing, it's contributing at least to some degree. It probably, and I'm sure, is not the whole story, but it is, it's in there as one of the mix of contributors. And the good news is that, or maybe the bad news, depending on the effects, but the good news is that it's certainly malleable. And the interesting unexplored thing, number one, is that it does interact with other things, given that depression and telomere shortness and outcomes for bladder cancer patients is just, I think, a first hint at that kind of thing. But I'm also very interested in, this is a really open question, in populations, are we, based on what's going on in our societies and our communities and our lives, are we handing on to our children shorter telomeres in ways that will then reverberate into population-sized or cohort-sized effects? Because those telomeres, we know they're handed on directly to children. And so the question is, do children recover if your mother gives you some very short telomeres? Can they recover or not from that? And so I think that raises me, really, really serious questions about the impact of what we do. And scientifically, it might account actually for some of the mysterious heritability of certain common diseases that had been hard to pin to genetic variants. But here's a way that you can get variation in disease risk through this particular mechanism. So again, I want to emphasize this is interacting with all sorts of other processes. It's by no means, telomeres are everything, but it's one thing that we can really study, and I think the questions are really very much open and certainly for me, raise larger issues than just biological or medical ones. So I'd like to say thank you very much for your attention and thank you for inviting me here.
I just would like to thank you and thank Elizabeth for a very inspirational talk and also for your generosity. And somehow I think it was, we learned a lot. We learned that it's not good to be stressed. And somehow we managed also not to get stressed because we have a remedy here in Switzerland that now we would like to share with you. Oh. It's chocolate, of course, that probably it's doing very good to telomeres.
And if it's backwards, it says desserts, right?
Yes.
Right. Thank you. How beautiful. Oh, my goodness.
Thank you very much. Thank you.
Thank you. Thank you very much. It's lovely. Thank you. Oh, I think we should put it above the flowers, don't you? Just the flowers are so sweet. One of those worked.
So only maybe 1 minute a comment. This is a chocolate with the logo of the Stress Network. And as Dominique mentioned at the beginning, it's a new network. We are the co-founders. I am Carmen Sandy from the EPFL, from the ETH Lausanne. And we saw that stress research is very important, and also that it has impact in society. So now we are very enthusiastic about continuing and be attuned. We have a website that you can consult. There will be some events in a couple of weeks during the Brain Week. There will be talks here on the topics of stress, etc. And you will be hearing more from us. But if you are interested in contributing or interacting with us, please contact Dominique or myself. And thank you very much. It was a great lecture. Thank you again, Elizabeth. Thank you.